Hi, Ricardo. Hi, everyone. It's a great pleasure today that I am interviewing longtime collaborator and friend, Ricardo Henriques. So I think we will start maybe by briefly presenting ourselves. And I will start first, and then I'll just shut up. I'm Christophe Leterrier. I am a team leader in Marseille, the Neurocyto Lab, and I work to decipher the organization of the cytoskeleton in neurons using a lot of advanced microscopies. And I guess this is the topic we will talk about today. So who are you, Ricardo?
Hi. First of all, before introducing myself, Christophe, thank you so much for taking the time to be in the interview. So I am Ricardo Henriques. I'm a group leader at Instituto Gulbenkian de Ciência, and I recently moved my lab from University College London and the Crick Institute in UK to IGC in Portugal. And as you said, Christophe, we've been colleagues and friends for many years. So it's actually ultra fun to have you here for this chat with me today.
All right. It is a lot of fun for me too. So that's a very brief introduction, and you know, this is about you today. So what can you tell us about exactly what you're doing as a researcher, and why you are interviewed by the RMS, and yeah, who you are as a scientist, and maybe who you are as a person too would be nice.
So as a scientist, I wear many different hats depending on the occasion. But I guess at the core, I can call myself a microscopist. And I am a microscopist from the perspective that I use microscopes to study cell biology. But I also develop technology for microscopes. And I guess that's one of the reasons why RMS has invited us to be in this interview. In microscopy for many years, I've specialized in single molecule supersolution microscopy. I'm actually a physicist and an engineer by training. I think you're also an engineer and no, you're a natural scientist by training, aren't you, Christophe?
I am an engineer by training, but you know, there are good ones.
but it's interesting because I always got interested in the aspect of pushing what we can do with microscopy, and when the field of supersolution microscopy started developing, I got FOMO and I wanted to be part of it. And to a large degree, it was on that stage that I also met you. You were at the same time trying to exploit supersolutions to ask quite interesting questions in neurosciences. At the time, I wasn't interested in a particular question in biology yet. I was more into the technology side of things. And so today,
yes.
Not only am I interested in supersolution, but I also use supersolution to study how viruses infect cells and how cells signal with each other. And I've also started doing a little bit of machine learning for bioimage analysis in parallel.
Okay, so maybe tell us more, like in practice, like, you know, where is your lab? How many people are there? How it's organized, and what you do? Maybe more. I mean, what kind of projects you have and who's doing that in the lab, you know, something like to get us a feeling of what it's like the everyday life of professional life of Ricardo Henriques.
So I like to think that we're an interesting lab from the perspective that we're a mix of researchers from fields of biology, physics, maths, and computational sciences. And we're interesting from the perspective that there's quite a diversity of backgrounds, and we all have to find a common language, which is one of the fun bits of the lab. And the Well, the main lab right now is situated in Portugal. We maintain connections to the UK, where there's still PhD students finishing their thesis. And the range of topics that we cover are a little bit on that directions, that we're using microscopy to study how HIV infects cells, and particularly undergoes uncoating and morphogenesis. We also keep on developing super resolution microscopy, both from the aspect of trying to make smart, inexpensive super resolution microscopes, and also algorithms that enable us to extract super resolution data from images. And we also exploit machine learning to do real-time analysis of microscopy data to classify events and discover components that are important for us in cell behavior in space and time. And one of the cool things is that I guess, like many labs, I don't see necessarily a physical boundary to our lab, because we have many collaborators like yourself. So you know, there's the people that I see every day. There's the people that I talk with almost every day via Twitter. and the, you know, like, I feel that there's a lab, and there's also a community around things that we like to do and that we contribute to and get contributions from, and I find that pretty cool.
Yeah, I think that's a very important point. I guess you know, the lab is not just the lab, but the lab is the lab, and it's your collaborator. And it's like, with today's communication capabilities, we can basically work with people from all around the world. And in particular, with the pandemic, it has taken a very important role, I guess, to you know, to have like to work with other people that are not physically here. I just would like to talk a bit more about the science for now. Like, so I think you coined, I don't know if you coined the term. I mean, you've been using it, at least, computational microscopy, right? So I saw that that's how you describe what you're doing, and I think it's a very interesting term. So could you tell us a bit more about that? Like, what is it exactly, computational microscopy? And also, what is needed now? I mean, why, what you say, like studying how HIV enters the cell, and you know, what do we need now that we don't already have? And why have the microscope to be made better? And in which way to be able to do exactly what we need to do for that?
So computational microscopy is pretty cool, because pretty much what we're talking about is that we found ways to extract more information beyond what we originally thought was the physics limits of a microscope. So we found computational ways to enhance resolution and to make a microscope go beyond what it was originally designed to. And from that perspective, almost everything that we do now involves, to some degree, having algorithms that are analyzing images to extract information beyond what is done classically in microscopy. An example is microscopy, but for example, now we're increasingly using event-driven microscopy. And by event-driven, it means that we actually have microscopes that in real time are able to characterize what they are observing, and then they're able to adapt the way that they acquire images based on what they are observing. And doing so, it maximizes the capture of the information that is really important. And these has several reasons. One of the key reasons is because microscopy is unfortunately destructive to living cells. They're really not designed to deal with the amount of light we use in a microscope. So we're trying to make intelligent microscopes that are able to reduce photo damage by only seeing what is needed when it is actually needed, and at the correct spatial and temporal resolution scales. And where are we going this? And where do I see the field going? Well, it's clear that almost everyone is trying, in some way or the other, trying to look into ways to build smart microscopes, right? And if you see developments like, for example, MinFlux, where you have ideas like single particle tracking in real time on the confocal microscope to increase resolution and minimize photo damage. The same way that you see super resolution algorithms becoming an integral part of microscopes, right? And now the computational side comes hand in hand with microscopy itself. I know that, for example, you've played with it. I haven't played it with it now, but it seems, for example, that even for companies like Nikon's, AI is becoming an integral part to their acquisition software. And I think that's quite fascinating. But a lot of this is born in the community and on open source algorithms. And it's the thing that gets me up at night and gets me really excited is that not only are we contributing a lot to that component where we're, as a community, pushing microscopy forward by making all these algorithms available and all these ideas transparent, but that I personally at my lab learns a lot from others. And look, it's beautiful because when I read a paper in biology, I go to the methods section, and often I get very little out of that. Normally, methods are extremely condensed on papers. It doesn't really allow me to see or understand how I can actually do those experiments. But code is functional. And functional code means that you know everything and you're able to build upon it. And I really like that size of microscopy right now.
Yeah, I think that's I think you're right at the a place where things are interesting right now, like this intersection between computation and biology, and it's quite fascinating to see this evolving. Let's take things back a bit. I would like you to tell us about how you ended up doing what you're doing and being who you're being. So can you tell us about what you did and your experience, PhD and postdocs? And I guess at some point you invented light sheet microscopy, so maybe you can tell us about that. And yeah, so let's hear your story.
Good, you're throwing off my balance.
So for the viewers, I must say this is a joke. He didn't invent light sheet microscopy.
It's a running joke between me and Christophe. All right, so how it started? It started by me being lazy. What I mean with that is, my dad is a mathematician and my older brother is a physicist.
So you're a failure in the family, right?
Yeah, I kind of I fell in love with many of the things that we were discussing on the table, you know. But at the same time, I kept a narrow mind because I was comfortable with what was being talked about in the dinner table, and that made me interested in physics, and particularly made me interested in particle and quantum physics at the start of my very, very early career. And then I noticed that I was wrong, that in reality, for me personally, at some point it became boring and became boring because I didn't see many practical applications on what I was studying. And then at the same time, I fell in love with a girl who was a microbiologist and a cell biologist. And naturally, we started talking about what we were doing in terms of studying.
It's a love story. I love that.
Yeah, and you know, the funny thing is when you start doing research, as you know, is at some point, suddenly all your friends are researchers also, or a large amount of researchers. And you're desperate trying to get experiments to work, and your friends are talking about the experiments to work. And then if you have a partner from biology, it's like suddenly your friends are a mix of the people from your field and that other field, and we're all talking to each other about how desperate we are to get data. And in the middle of it, I'm listening, I'm like, what they're doing is pretty cool. But the funniest thing was to get that moment where you go like, it's pretty cool, and I have a different perspective on how it can be solved. And that's the thing, right? That was the moment that, you know, researchers counting cells by hand on a microscope with a counter, 1, 2, 3, and they were like, I can pretty much do an algorithm to solve that in 3 seconds. And I started realizing that, you know, that here's something that I could get a lot of fulfillment from, and that I could perhaps find a different angle to go about it. So that ended up taking me in that direction. And so I ended up moving from particle physics into a cell biology institute. It was embarrassing at the beginning, embarrassing from this perspective that it's a field where I didn't study too. I had practical training in biophysics at the university level, but nothing that would allow me or prepare me to actually be in the middle of a cell biology lab. But the beauty of it is that you learn from osmosis. I guess it's really easy to get someone from a different field that is not cell biology and put them among cell biologists, and they will learn. That it is for a cell biologist that is in a cell biology lab to suddenly say, I'm going to study physics. And naturally, that just helped me adapt to the field, bring something from where I come from and from what I like, physics, computational sciences, into biology at the same time, start to understand what interesting questions I'd like to try to answer from there. And I get easily excited, as you know.
I know that.
So whenever I see someone doing something cool, it kind of primes me to becoming more interested in that direction.
Where was that? I mean, because there's something about you that is quite difficult to understand, is where you were working during your various stages of your career. So now you're talking about things in Portugal, right?
Yeah.
So what happened next? How did you end up doing your PhD between South Africa and Paris? That's what I want to hear.
Good question. I trained as a physicist in Portugal, right? I wanted to go into cell biology, but particularly I wanted to go into artificial vision, which at the time is what we would call bioimage analysis. Okay, and there was nothing that I believe there was nothing in that direction in Portugal. So I actually started doing a PhD in robotics. Because from robotics I could get artificial vision, and from the artificial vision I could apply it to do bioimage analysis and go into microscopy. And that didn't pan out, and they didn't pan out because that was a field that didn't exist in Portugal. I couldn't really get the knowledge and resources easily from others to become a leading expert in that area. And at the same time, that I was, a little bit desperate about trying to push that PhD in robotics into the direction I wanted. A new lab in Portugal appeared, that's Musa Mhlanga, who became my new PhD supervisor. And I actually stopped my previous PhD and started a new PhD with Musa from scratch. And that was a PhD, with already exploring the ideas of super resolution microscopy and trying to understand how you get gene transcription bursts by using super resolution on yeast cells. And the funny thing is, sneakily, Musa got a double appointment, as in he got an invitation to also start a lab in South Africa. And so the expectation was that he would spend a large part of his time in Portugal and a large part of his time in South Africa. And I thought, okay, I'll be his Portuguese lab. But for personal reasons, he ended up spending most of his time in South Africa. And also in Portugal, I was striving to get that knowledge on bioimage analysis and computational biology and optics that I didn't have locally. So one agreement that we made is I could visit some collaborators that were starting to build super resolution microscopes in Paris. And at the time that was Christophe Zimmer and Maxime Dahan. And also Jean-Christophe Olive-Marin was doing bioimage analysis in the terms that was interesting to me. So I went to
Pasteur Institute.
I went at Pasteur Institute. So I went to visit for 3 months. And then those 3 months became 6 months. And then those 6 months became half of my PhD. And then the other half, it was me visiting South Africa, where I built a super resolution microscope and did a little bit of research with Musa, and then me jumping around a little bit into Portugal, a little bit into the US, where suddenly I started also consulting for Andor Technology, a microscope development, I guess more for camera development company. And Belfast, where they had the factory with Andor. So I was jumping around all through these places. And that was confusing. And for a long time, I felt confused. But then when I finished my PhD, I went like, that was awesome. That I could actually cooperate with so many people and experience so many different communities while building my PhD. And I really enjoyed staying with doing research with Christophe Zimmer, and Christophe asked me if I'd like to stay for a little bit longer in his lab, and I accepted then to do a postdoc with him. That got me the chance to use the microscopes that we already had in the lab, plus some of the tech I created during my PhD, and then apply it to start studying T-cell signaling and a little bit of HIV infection also for the first time. One of the nice things is that from the ground up, when I started doing my postdoc, I told Christophe that sooner than later, I would be looking into starting my own lab if I had that chance. And he was incredibly supportive of that. So I was actually able to do a very short postdoc. I think it was a year and a half or something like that at the end. And then I had the opportunity to start a lab in the UK. There is one trick here, which is the reason why it was such a short postdoc and why I was able to jump was also because I realized that it was a really special moment in time, because super resolution microscopes were about to become commercial. And I realized that with those microscopes becoming commercially available, there would be many research institutes that would look into purchasing those microscopes and equipping themselves with super resolution. And they would lack local expertise to exploit those systems for the first time. As you know, a single molecule super resolution microscope, it's a little bit like an airplane. You can buy an airplane, but you still need someone with hours of flight experience to fly that airplane.
Really?
And so it's just not about getting those microscopes. You need people that know how to drive those microscopes. And I was expecting that, right? I was expecting for that opportunity, and that opportunity arose, and it helped me very quickly establish a lab. Light sheet microscopy.
When was that? You started your lab in lab?
So 2013, I started the lab, and that was a direct move from Paris to London, which is super cool because that just means taking the TGV, the Eurostar, between Paris and St. Pancras in London.
Yeah.
So that was
I remember when we met. I mean, it was later than that. I think 2015 or 16 when we met on Twitter. I think we can say that. I can tell that you sent me a message on Twitter, and that's how it started. And we met later at the single molecule localization microscopy symposium, I think, in real life for the first time. Yeah, so it was like 2016, and I was trying to start my lab, and I saw you, and you had your PhD like 5 years after me, and you had your life for 5 years. I'm like, how did you pull it out? So, yeah. So you started your lab in 2013, and when we started to work together in 2015-16, it was already quite successful, and you were doing stuff, of course, in single molecule localization microscopy, but you were already, as I recall, transitioning to this computational microscopy approach with the SURF. Basically, SURF was your first work in that area. So yeah, maybe tell us a bit about the road from SURF to Squirrel to things that I know personally a bit better, but I'm sure people will be interested in.
So at some point I backtracked. I think it was, when was it? I think it was when I at some point introduced you on one of your talks. I backtracked the moment that we first talked.
Yeah, I think you found the email or the message.
And it was it was an email about QuickPalm.
It was about Squirrel. I think it was just after SURF was out. And you were working on Squirrel, and you wanted
the first email was like 2008, and it was you on the forum.
Oh yeah, that was
on my forum.
Yeah. Sorry.
Asking nicely if the drift correction didn't work or if you were doing something wrong. And in fact, the drift correction was sucked on QuickPalm at the time.
Yeah.
And then
we're talking Google code era for the people who weren't there.
And then I kept an eye on you on Twitter. At times that I wasn't known a lot. Let's put it that way. So SURF, right? SURF, I guess it's one of the main reasons why I got some recognition. Well, QuickPalm was the first thing that got me recognition, and then the SURF algorithm. And the funny thing is that SURF is kind of QuickPalm too. What I mean by that is that so QuickPalm is like this algorithm that I developed in, I'm going to say 2008, I might be wrong. No, 2010 was when it was published, I think. And it was one of the first open source single molecule localization microscopy algorithms, at least one that became, for a little bit of time, the most popular one, I would say. And then throughout, and then I did that during my PhD and throughout my postdoc, I kept on developing QuickPalm, but I didn't release it because I didn't felt that it was significantly different and ready yet to the community. And then I started the lab in 2013, and I was kind of, I kind of convinced a couple of my researchers, Nils and Sean, to work around some of the ideas I wanted to build on around QuickBound. And so Nils started looking into SOFI and how SOFI could be adapted to use single molecule localization data. And Sean was working on the single molecularization itself, particularly using concepts from radial symmetry. And these were actually two projects that at some point got merged together, and then SURF appears out of it. And when we got SURF out, SURF, there's a regime of data where it does a very good job, and there's a regime of data where it does not at all, right? It's meant to be a very good high density single molecule reconstruction algorithm. And at times, researchers were a bit naive in the way that they were using SURF, and their data was full of defects that were problematic if you started extracting some biological meaning out of that. And that's a trap that it's easy to fall into. And I personally have fallen into that trap for some other reasons throughout my career when I was doing single molecule microscopy. And at times you get artifacts that are so visually appealing that you think there's actually a biological structure there when there isn't. So we wanted to work on something that would help guide researchers to understand if there was problems with the data that they were getting out of SURF, but also out of almost every other superresolution technique, because this is not a problem particularly to SURF. If you have the wrong input data to some of these superresolution approaches, you're going to get defects out of it, and those defects might be visually appealing enough that you think there's biological meaning behind them. So that's what got us to develop Squirrel, right? An algorithm that was able to map some of the errors and defects and artifacts you get out of superresolution data. And it was when we started going through that idea, we thought, or I thought, I should say, my data is at times okay, but it dominantly sucks. Who can I work with that has some of the best data I've ever seen? And that's pretty much, it took some courage to drop you a message, because some of the data that you were acquiring and you acquire every day, it's freaking amazing, Christophe. And I was going like, he's not going to answer me. And the fun thing is that I remember I was completely surprised, right? Because we had a chat, and then the next day you were acquiring the data we had talked about, and I was going like, I was expecting, either to be ignored or maybe in 6 months we'll start actually getting some correlation. And in a few hours, we were talking about how to build that paper up. And that's pretty cool. So that's how we ended up developing Squirrel, but also I guess the beginning of us working together. And what at the beginning, I don't remember how many papers we have together meanwhile, but if we're not in double digit in a few short years, we might get into double digit.
I think we are soon. But I remember that exactly this moment, and I have to say from my side, things were. So I was starting my lab, and I had spent so many years in front of those microscopes that yes, I knew how to do good images, and you know how it is when you do biology, because what I do is use these microscopes to do biology for looking at some problems that I try to solve. But you acquire so much data that ends up not being used just because you are trying, okay, maybe if I change the buffer a little bit, what does it do? So you take those cost cells and you do a standard microtubule labeling, or you optimize your immuno. So you try 10 different ways of staining microtubules, and you do that in regular cells. So in the end, this will never be in any paper, because in any neuroscience paper, because nobody cares about that. So when you contact me, I was like, okay, maybe all this data that is useless as a material for building results, it's just, you know, training or for us to get better or for us to optimize things, can actually be useful. And I thought, you know, in the same way we try not to use animals for nothing, or to try to refine their use so that we are as efficient as possible. I think it's the same with data. It's a very good thing that any data you acquire, you try to do something with it. And the fact to be able to connect with you, and that you would always be happy to have data and to do something with it and to try to make it better, not by just, you know, trying 10 different buffers, but by using computation. I think this was a very important and good opportunity for me to actually make it usable and make it efficient. So that's the main reason why I was very happy. And you came early enough so that you know, I was very happy to do it with you.
You still had time.
Yeah. So yeah, and I think in the end, it's been really wonderful for both of us, because there is this complementarity that I always have some data that is either very good or subpar, and you can work on both and try to see what you can do for the subpar to be better, or how you can use the good one to make the other better. So there's always something to be done on that front. So I think that's a very fruitful collaboration. So we were at Squirrel, so of course, and we've done thousands of papers together since. But let's fast forward to today. You are in Portugal. You recently relocated your lab, and you mentioned that you also had a kind of thematic movement and evolution toward more machine learning. And I wanted to, because I think a lot of people either are into this or are looking at that from the side, you know, like, okay, machine learning, everyone is about deep learning. And you know, so there's a big enthusiasm about it, or some people on the contrary are like looking and say, no, you know, you don't know what you do when you do that. So I remember I just want to mention at the first time I actually discussed that with you, I think it was in London in 2017, and you were working together with the people that were doing the CARE algorithm, which was one of the first use of machine learning to get better microscope images. And I was completely naive about it, and I asked, but what can you do that you don't already know if you just train on things you have already seen? And I remember you managed to explain it to me in such a way that I actually still use the explanation you gave me at the time when people asked me the same exact question. Like, it's all about exploiting the redundancy that you cannot see by eye, and that something is there that you can take advantage of, and you don't see it, but the machine will see it. So I think this tells us how mature you were already on the subject back then, and so can you tell us a bit more about how you use these new technologies such as deep learning to make microscopy better?
Yeah, so there's been a central theme almost on everything that we do, and that central theme is really simple, which is making new technology accessible to the field of biology. And what I mean with that is, I grew up looking at papers from optical development labs and computational labs that they were building this beautiful technology, but they put it out there in the form that the only researchers that could exploit it would be their collaborators or that lab themselves. And I made my career of trying to fight that, trying to go the extra mile and either develop technology or grab pre-existing technology and make it adapted in the form that a non-experts could immediately exploit it to their own research. And so our first step in machine learning was kind of almost by accident. Although I was really curious about it, I understood that it was going to be revolutionary in the way that we do research in all fields. But there wasn't immediate, it was so early on that I didn't have any immediate reasons to touch it. And it was the team behind CARE, and at the time, because I had a connection with Leterrier, and through Luik, then Martin and Florian, they reached out because they wanted to add a super-resolution component to CARE, and they invited me to get into it. And I was, super happy to be able to contribute towards that project, and my team also. But the bug of it remains, and the bug was there. Machine learning is super cool, but it's not really accessible. It's like if you're a cell biology lab and you want to use it, you need to cooperate with another lab or hire a postdoc in computational sciences. And I started in my mind thinking about how we could make it easier. And there's a few people out there that were also considering the same thing. That's one of the reasons why, for example, we started collaborating with Guillaume Jacquemet, because some of these things, which is fantastic, right? You know about all this because many of these things started on Twitter, right? With us going like, hey, we're using Google Colab to run some of these algorithms. Here's without a paper yet. Here's a notebook with some of the things that I am trying right now. And then Guillaume goes like, I'll try it tomorrow. And you see that coming from there, and suddenly you start saying, okay, it's like there's a community building around this, and there's some ideas there I want to exploit, and it's easy to build up on those collaborations. That's how we started collaborating years before on super resolution, because we were on Twitter discussing some of these ideas that ended up becoming papers that we got together. So right now, what we've done with machine learning was that focusing on making it more accessible. What we're going to do in the future is actually trying to use machine learning on a microscope to try to make the microscope make decisions. Do I need to take an image or not? Because it's damaging, it's because you need to prioritize where do you want to acquire an image, because you can have massive samples with small fields of view, and we might have completely new ideas that we contribute there. We might not. There's many people might be thinking on some of these directions. What I can promise you is that what we'll do will be immediately accessible and available and easy to use to everyone else, because that's our ethos at the end of the day in the lab.
That's a very important point, and maybe that's the point where I would like to discuss a bit before we finish. It's so you are very much involved in let's say open science, but I think it's more than that, because the way you work and the way I've worked with you and I've seen you working with others is, as you described, this very organic way of being, okay, I'm doing that, do you want to try? But I'm also trying this other thing, okay, let's do it together. Something that is not only about publishing preprints or stuff like that, but really a way of making science that is intrinsically collaborative, and that's I think is also very a lot of fun. And so I would try to merge those two aspects. Like there is the open science, like more classical open science approach, like we want to make things available, we want to have the software easy to use, and we want to know people exactly what we do and when we do it and make them accessible. And I think zero cost is really the epitome of that approach. Like you have brought the possibility to very simply use deep learning for everyone with this work. And also it's connected also to the use of social media and to the use of like being a community. And I think this is an important aspect that I would like to have some thought about, like doing science like you're part of a community and in a very distributed way that is allowed now with the things that we are doing computational work with electronic devices. So basically there's no reason not to do it all together at the same time. So what do you think about that?
It's very interesting because the way I classically look at the way research is done is that it's extremely hierarchical. What I mean with that is there's kind of this currency, which are publications or grants. There are also different forms of currency, and labs try to hoard their currency and compete with each other. And the institutions will look at the value of researchers depending on how much currency they have, how many papers, where are those papers published, and this kind of seems very narrow focused. I almost think that the way we tend to do research is the way that millionaires behave. They try to hoard their money and to evade taxes, which will be the equivalent to find weird ways to get those big name papers out and to rise up in their institutions. And there's other ways to do it, right? There's the way that it takes a little bit of courage, but you can almost go like, screw it. I'm going to be as open as I can about what I want to do because I'm really excited. And I'm going to tell those people that what they're doing is exciting because I feel there is exciting. I'm going to tell about what I'm doing right now because that's who I am. And I'll invite researchers to help me out, or I'll ask, do you need a hand? And it's interesting. It's breaking that economical size of research, right? It's a little bit like Web3 cryptocurrency, where you go like, let's decentralize, let's give the power back to the people. They can be their own directors of the way that they use this currency. And I see preprints as being that. I see Twitter as being that. You have a cool image, put it on Twitter. It might be in a preprint in 2 months, and it might be in a paper in a year. Does anyone care about IP protection, about those figures? No, because we're pushing the publishing houses to break that apart. So what does that allow me to do? It allows me to do research much faster than I could ever imagine before, because everything is crowdsourced, right? I see a bright new idea coming from our side or from another side. I go to social media, I discuss it with you. We go like, that sucks, or that's beautiful. And then suddenly that just generates into new ideas and new projects. And suddenly you start building a community around you, and you start building up in direction of getting something out. And the something else, will be out in many forms. It will be out on contributions to other projects. It will be out as papers. It will be out as technology. It will be out as resources. And all this, the nice thing is that they're all by definition shared. And there's nothing there that is about, let's keep this a secret just for us to build up citations and build up our capacity to have that job on pristine institute at the end. This, of course, has the downside to it, right? It's different from the way that some researchers like to do things, and I understand it, and I at times see pushed back from that. But I think the way that we're doing things, and social media is definitely helping on that direction. I think the way that we're doing things and exposing and being open and picking up on each other's capacity and know-how using platforms like Twitter is, if not the way to do things in the future, I think it's going to be a big component on how we're doing things now and how we're going to likely improve on the way that we do science in the future. What's your take on it, Christophe? By the way, I'd be keen to hear.
I think you summarized well the issue, and I agree with you. I think this is actually one of the things I learned from you, that it's always better to be open, and it's been, for me, I think a lot of good things happened to me because I was always open to new things and to work with people and to, you know, and not to be defensive, and so I'm not saying that it works for everyone, and that I mean sometimes I also have the feeling that maybe the next step to this kind of whole, okay, let's be open, let's discuss everything on social media, let's do the science all together. We have to take to be aware of biases and of the fact that some people are left behind, and that some people don't have a voice in the global conversation and in the community. And I think that's something we have to work on. But I would say that is maybe the challenge of this kind of way of doing things, which can be really like, you know, you hear the people that are the louder. So this is something that has been to be taken into account. But it's always much better, infinitely better than the old way, where there wasn't any communication. So this is for this. I mean, it's clear that I'm very excited about this new way of doing things, and I hope to be a small part of this movement with you.
Good. I completely agree, Christophe.
All right, so let's wrap up. Last question, maybe. So kind of cliche question, but what advice would you give to a young researcher that is starting today, or yourself a few years or many years ago? I don't know how we can consider ourselves old or not.
That's an interesting question. Weirdly enough, I at times think that every time that I was shown a barrier in front of me, it's being, you know, another researcher saying what you're doing will not go pan out, or it's too mainstream, or getting a paper rejected, or not being able to simply get something done. That has been more often more motivating than getting a positive answer and praise for something. But it takes it toll, as in it takes a little bit of you. It breaks you down a little bit. And if there's anything I would advise myself, if I could go back, is all those hard times where I had to face a barrier, and those barriers were important. I wouldn't take it back. The only thing I wish someone had said at the time is, "These shall pass." What you feel right now, and all these problems, it's all a question of time. These shall pass, and it's something that you learn with maturity, and I know, and probably you know, and these things now touch you much less than they touched you before, but the beginning, it's really, really rough. And I just wish that, I could go back and talk to myself, or I just had someone saying, it's just a stepping stone. Don't worry. you will do increasingly better, and all this will just be small bricks in the road.
Yeah. Okay, so I guess the advice is don't forget the big picture and keep on pushing forward.
Yes.
All right, I think that's a really good advice. Thank you very much, Ricardo, and I was very
happy Christophe,
to have the chance to have this little chat, and we can go back to making jokes on Twitter together. So if you can join us, thank you very much.
Thank you, Christophe.